Welcome to the PubMed Daily Review for April 01 SD. Today we scanned 4,596 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5. First human cell-based cultivation system for the syphilis spirochete Treponema pallidum by Uri Bozak and David Smages from Biology, Faculty of Medicine, Masaryk University, Brno, Czech Republic. Let's break down its key contributions. Syphilis, caused by Treponema pallidum, remains a significant global health concern with increasing case numbers. For decades, researchers have struggled to grow this bacterium outside of living hosts, primarily relying on laboratory rabbits. While a rabbit cell-based system was developed in 2018, a truly humanized system is needed to unlock new avenues for studying how this pathogen interacts with human cells. To address this, the researchers tested 3 different human foreskin fibroblast cell lines, comparing their ability to support T. pallidum growth against the established rabbit epithelial cell line. They used 8 different strains of the bacteria and monitored their growth over a 10-week period using microscopy. The core idea was to see if these human cells could effectively serve as a host for the syphilis spirochete. The results were promising. All 3 human fibroblast cell lines were able to support the growth of T. pallidum. Notably, the DAL1 strain was continuously cultivated on human cells for over a year, demonstrating the system's long-term viability. While growth was generally slower compared to the rabbit cell system, with generation times ranging from about 39 to 55 hours on human cells versus 33 hours on rabbit cells, the system proved capable of supporting 8 different strains, including both Nichols-like and SS14-like clusters. This study represents a significant step forward by establishing a human cell-based cultivation system for Treponema pallidum. This humanized model opens exciting possibilities for studying syphilis pathogenesis and host immune responses in a more relevant context. While further optimization might be needed to match the growth rates seen in rabbit systems, this development is crucial for advancing research towards new diagnostics and treatments for this persistent global health threat. Up next, our second paper out of 5. Large language model detects previously undiagnosed heart failure with preserved ejection fraction in patients with metabolic-associated fatty liver disease. A multicenter cohort study by Xiaodan Liu and Thomas Kisimbi from Department of Gastroenterology, Hunan Provincial People's Hospital, Zhengzhou University People's Hospital, Zhengzhou, China. Let's break down its key contributions. Heart failure with preserved ejection fraction, or HFpEF, is a growing problem, but it's notoriously tricky to diagnose. Symptoms can be vague, and the necessary information is often scattered across patient records. This is especially true for individuals with metabolic-associated fatty liver disease, or MAFLD, who are already at higher risk for heart problems. We need better ways to find these patients early. To tackle this, researchers used a specialized large language model called MedGuide 14B. They applied it to the electronic health records of over 24,000 patients diagnosed with MAFLD. The model analyzed both structured data and clinical notes to identify potential HFpEF cases that hadn't been clinically recognized. These model-identified cases were then compared to those already diagnosed by doctors and to patients without HFpEF, looking at outcomes like hospitalizations and survival. The results were striking. MedGuide 14B identified over 4,200 previously undiagnosed cases of HFpEF, representing an additional 17.6% of the MAFLD cohort. The model demonstrated excellent performance, with an AUC of 0.94, a sensitivity of 95%, and specificity of 92.3%. Importantly, over 90% of these newly identified cases were confirmed upon blinded review. These patients, while having lower risks than clinically diagnosed HFpEF, still faced significantly higher rates of rehospitalization and mortality compared to those without HFpEF. This study highlights the powerful potential of large language models to revolutionize disease detection. By leveraging routine electronic health records, MedGuide 14B significantly improved the identification of HFpEF in patients with MAFLD, a group often overlooked. This opens doors for earlier interventions and better patient outcomes. While promising, further prospective studies are essential to confirm these findings across different healthcare settings before this technology can be fully integrated into clinical practice. Up next, our third paper out of 5. CD8 term-like T cells expressing perforin and IFN gamma define a pediatric activated T cell acute liver failure endotype by Tamir Diamond and Edward M. Behrens from Division of Gastroenterology, Hepatology and Nutrition, Children's Hospital of Philadelphia, and Perelman School of Medicine at the University of Pennsylvania, Philadelphia, Pennsylvania, USA. Let's break down its key contributions. Pediatric acute liver failure is a serious condition with high mortality. A significant portion of these cases are driven by an overactive immune response, specifically involving T cells. We call this activated T cell pediatric acute liver failure, or TCPLF. Understanding the specific immune signatures within TCPLF is crucial for developing better treatments and improving outcomes for these children. To investigate the immune landscape of TCPLF, we analyzed samples from children enrolled in the TRIUMPH trial. We compared patients with TCPLF to healthy children and those with other forms of liver failure or related conditions. Using advanced techniques like flow cytometry and single-cell RNA sequencing, we examined their peripheral blood cells and serum. This allowed us to identify distinct immune profiles within the TCPLF group. Our analysis revealed two distinct endotypes within TCPLF. One group, termed TCPLF high PRF, showed a significantly higher percentage of perforin 1 expressing CD8 plus T cells. These patients also had monocytosis and a unique circulating CD8 plus T cell population with markers of tissue-resident memory and IFN production. The other group, TCPAF NL-PRF, exhibited hypergammaglobulinemia and increased B cells. Transcriptional analysis further supported these findings, showing enhanced cytolytic activity in the CD8 plus T cells of the high PRF group. This study significantly advances our understanding of TCPLF by identifying distinct immune endotypes. The high PRF endotype, with its specific CD8 plus T cell signature, presents a potential target for novel immunotherapies. Recognizing these differences is crucial for tailoring future treatments and improving transplant-free survival in children with this severe condition. Future studies should focus on validating these findings and developing targeted interventions for each endotype. Up next, our fourth paper out of 5, Emergent Critical Oscillations in Motor Cortex of Parkinson's Patients by Chung Lai and Woodrow L. Hsu. Let's break down its key contributions. Brain function is believed to be optimal when neural activity operates near a state called criticality. This state, poised between order and chaos, allows the brain to be flexible and process information efficiently. While criticality is observed in healthy brains, it's often disrupted in various neurological disorders. This study investigates whether Parkinson's disease also leads to a deviation from this crucial dynamical state in the motor cortex. To investigate this, researchers analyzed resting state electroencephalography, or EEG, data from the primary motor cortex of individuals with Parkinson's disease and compared it to healthy controls. They employed advanced analytical techniques, including information theory and temporal renormalization group theory, to measure proximity to criticality. The study focused on identifying emergent oscillatory patterns and assessing their dynamical state. The study found that individuals with Parkinson's disease exhibited prominent oscillatory brain activity in both low delta and high theta frequency bands, which were not observed in healthy controls. Surprisingly, these emergent oscillations were found to be close to criticality. Both established and novel analytical approaches confirmed this finding, demonstrating that critical dynamics can indeed be associated with a disease state, not just healthy brain function. This research significantly shifts our understanding by demonstrating that criticality isn't exclusively a hallmark of healthy brain function. It can also emerge in disease states like Parkinson's. The presence of these near-critical oscillations in the motor cortex may offer novel avenues for understanding the underlying mechanisms of motor impairments in Parkinson's disease. Future studies should explore the functional implications of these findings and whether they can be leveraged for therapeutic interventions. Up next, our fifth paper out of 5, CRISPR and the Promise of Gene Editing. Unlocking Curative Possibilities for Liver Disorders Worldwide by Sabah Kulsum and Dushyant Singh Dehia from Department of Internal Medicine, New York Medical College. St. Mary's St. Clair's, New York, New York. Let's break down its key contributions. Liver diseases represent a major global health challenge, causing millions of deaths annually. While treatments have improved, many conditions remain chronic and only partially treatable. The dream of a cure is now within reach thanks to CRISPR gene editing, a technology that could fundamentally rewrite the genetic codes driving these diseases. This paper reviews the exciting potential of CRISPR gene editing for liver disorders. It examines how CRISPR-Cas systems are being applied to monogenic metabolic diseases, chronic viral hepatitis, and hepatocellular carcinoma. Early successes, like the treatment of transthyretin amyloidosis, demonstrate the power of in vivo gene editing to achieve meaningful clinical outcomes. A major milestone was the intravenous administration of a CRISPR therapy for transthyretin amyloidosis, resulting in a dramatic and sustained reduction in TTR levels. This proved that in vivo gene editing can yield clinical benefits. While chronic hepatitis B affects nearly 257 million globally, and liver cancer is a growing threat, CRISPR offers novel strategies to target these conditions. CRISPR gene editing represents a profound shift, moving us towards curative therapies for liver diseases. However, significant ethical, safety, and access challenges must be addressed. Continued research, collaboration, and careful regulation will be crucial to responsibly harness this technology for global liver health.